So in this part of presentation we will see the promoter sequence and also we will discuss about RNA polymerase enzyme. Okay, fine. So the question is, what is a promoter sequence, right? So the promoter sequences are certain consensus sequences or conserved sequences seen along with most of the or all the genes seen in prokaryotes as well as in eukaryotes. So these are certain sequences which are found along with the gene, right? And what is the significance? The significance is that the promoter sequences will identify or will help the system to identify a gene. Clear? So without a promoter, the gene will not be recognized by the machineries like the RNA polymerase or the transcription factors. That identification will not be possible by these proteins that this is a gene, right? Already we have seen that there are a number of genes in eukaryotic system and also in prokaryotic system. When you take an example in human cells, we know that there are 20 to 25,000 number of genes which are spanning in the entire chromosome. That means in the 23 set of chromosomes, right? But which doesn't mean that the entire DNA or the entire chromosomes are carrying the gene. It is not like that, right? When you see chromosome 1, for example, maybe there will be a number of genes, maybe 1,000 gene is there in the, for example, 1,000 gene is there in chromosome 1. And when you take chromosome 5, there is a 700 gene. In chromosome 10, you can see 1,100 gene. So it's so. So the rest of the part, that means within a chromosome or within when you take as a whole of the total chromosome, only 1.5 to 2% of the DNA in terms of its sequence carry a valid information. That means only 2% of the chromosome is usually considered as a gene. So the rest of the sequences are maybe non-coding regions. They will have certain other functions, but as of now no other vital functions have been identified. They are usually considered as the some other sequences which are involved in certain other mechanisms, but normally not usually transcribed, right? So these sequences are usually identified by the system, or the genes are identified by the system through the presence of a promoter sequences. Clear? So we'll move into that discussion along with a discussion with RNA polymerase enzyme. So this represents a promoter sequence in the bacterial genes. The two important sequences in bacteria which are commonly found are at locations -10 and at -35 position, right? And the numbers are given in reference to the +1 site, which is the start point of transcription. So before the transcription start point, 10 base pairs away from the start site, you can see a consensus sequence. Usually that sequence is called Pribno box, and the sequence is TATAAT. This is a conserved sequence. That means these sequences are found in majority of the prokaryotic genes in different bacteria, right? And the second one is at -35 position with a sequence called TTGACA, right? So these are the consensus sequences usually seen in prokaryotic promoter, and these are important for the gene to be recognized by the RNA polymerases and also the transcription factor for the transcription, right? And here another factor is also introduced, that is what we call the up element. These are some additional promoter sequences usually which will have some additional effect on transcription initiation, right? So whenever we have a multiple number of promoter sequences, the capacity of the gene to get transcribed also increases, and accordingly we call them as a strong promoter. And if the number of or the conserved sequences and also the number of sequences are less, then they are usually transcribed less frequently, and hence we call them as weak promoters, right? So in short, in bacteria now we have discussed at 1 at -10, 1 at -35, and one still upstream to the -35, which is located and that sequence is called up element, right? And this is what we see in eukaryotic system. So this is a eukaryotic polymerase 2 core promoter, which means that we'll discuss that later, that in eukaryotes we have different polymerases and also we have different promoter sequences also. So this is a sequence for the polymerase 2 enzyme, right? So these are a concise image in which you can see the most significant promoter sequences, one at +1 site or spanning at +1 site, usually called as INR sequence or the initiator sequence, right? And there are two sequences in the downstream and two in the upstream, right? The upstream sequences, one is usually located at -25 position, which is called the Tata box. They are called Tata box because of their sequences of TA, TA, AA or AT, right? The location is -25. And the second one is BRE, or it is otherwise the expansion is like TF2B recognition element. Clear? And the importance of the Tata box and BRE sequences we'll see in the coming discussions, right? And the other one in the downstream, one is the DPE or the downstream promoter sequences, and the second one is MTE, motif 10 element. So altogether this represent a eukaryotic promoter sequence in majority of the genes, right? And occasionally you cannot see all the genes, all these sequences in all the genes, but at least one or two will be present, and most of the genes carry the sequence called the Tata box, right? It's approximately it's around 32% of the genes carry the Tata box, right? So this represents the promoter sequences for eukaryotic system. Clear? So now moving into the third part, that's about the RNA polymerase enzyme, which is the enzyme involved in the transcription mechanism, right? It's otherwise called DNA dependent RNA polymerase enzyme. It was introduced in, or that was discovered in 1960s by science research groups, and as for many other things, it was initially discovered in bacteria. Then there are many things about this enzyme. I'm not going into the details much. In E. coli cell or in any other systems, you can see a number of such enzymes usually present in the cytoplasm, right? And the most important factor, already we have seen many differences in prokaryotes and eukaryotes, and one of the most important difference of RNA polymerase from that of a similar enzyme called DNA polymerase, which is involved in replication, is that this enzyme does not require a primer. That means without a primer, this enzyme can go for addition of nucleotide into a free 3' OH. That means which is the same reaction as that of a DNA polymerase. The only difference is that the first base also could be added by this RNA polymerase enzyme, which is unique to this enzyme, and that could not be done by DNA polymerase, right? So this is the mechanism. You can see there is a reaction in which a phosphate group is added into the OH group to form addition of a nucleotide, right? So in bacteria, as already said, we have a single RNA polymerase, while in eukaryotic system we have three sets, polymerase 1, 2 and 3, right? The polymerase 1 in eukaryote is exclusively for the synthesis of rRNA molecule, right? And they are usually localized in the nucleolus of the nucleus. The second one, RNA polymerase 2, is for the mRNA synthesis, and we are much interested on RNA polymerase 2 and mRNA synthesis, and we will be discussing in detail about this RNA polymerase 2 later. And the RNA polymerase 3 for tRNA and certain minor rRNA molecules like 5S rRNA and also some certain small nuclear rRNA molecules, right? So these are the classification of RNA molecule in eukaryotic system. And when we go to the structural details, it's a heavy molecule with multiple subunits of around 450 kilodalton, and there are a total of there is a 6 subunits, 2 alpha, 2 beta, 1 omega, and an additional sigma subunit, right? So the holoenzyme consists of a core enzyme plus a sigma factor, right? So the sigma factor is an additional factor. Clear? So the sigma factor comes to the core enzyme to become a holoenzyme. Even without the sigma factor, the enzyme is active. That means the enzyme can do their catalytic activity. But the sigma factor is important, we will see that. So these are certain comparison of the structural details. You can see homologous regions are present in all the systems like archaea systems and eukaryotes. You can see there the beta dash beta alpha and other subunits, and the comparative or homologous subunits in eukaryotic system and archaebacterial system, right? And of which the beta dash subunit is important, just like the RPB1 subunit of RNA polymerase 2. That means these are the largest subunit of the RNA polymerase enzyme, and which has a specific carboxy terminal domain, and these carboxy terminal domains will undergo phosphorylation during the elongation process, right? So these are the comparative structural details. And when we see some in-depth structural features of the RNA polymerase in E. coli, you can see the alpha subunit is usually required for the assembly of the enzyme complex. Beta and beta dash are usually acting as the pincers. That means they usually do the unwinding and opening of the helix. Then the omega again help in the assembly, but usually they are not vital, otherwise no other vital functions. And the sigma factor is the most crucial element, even though this is not part of the core enzyme. These factors are required for the enzyme to bound over the promoter sequence precisely, right? So without a sigma factor, the enzyme cannot initiate transcription exactly in the gene site, right? So sigma factor is very crucial for directing the enzyme to the core promoter region or to the gene itself. The sigma factor is like, this is the major sigma factor or major transcription factor in prokaryote. Already we have seen that in eukaryotic system we can find a number of such transcription factors. We call them like transcription factor 2B, 2A, E, F, like that. We have a lot of sequences. We have a lot of such factors in eukaryotic system. But in bacteria, we have only one major factor called the sigma factor, and which is involved in the recognition of the gene promoter, as already said. And we have different sigma factors, in bacteria, and they are categorized based on their function and molecular weight, right? And based on molecular weight, you can see like sigma 70 with 70 kilodalton molecular weight, sigma 32 28, 38, 54, etc., etc. So all these are exclusively working for different set of genes, not for the same genes, of which the sigma 70 is the housekeeping transcription factor in prokaryotic system. Right, the rest of them will have different functions, like you can see in that table. For example, sigma 54 is involved in the nitrogen metabolism genes. That means when the bacteria want to express the genes involved in nitrogen fixation, then the sigma factor for the identification of that gene is usually sigma 54, not sigma 70. Right, so that is the importance for the sigma factor. Right, so that's about these discussions. So I'll brief it. See, what we have seen now is we have a DNA sequence and we have the +1 site from where we have the transcription, and this is what we usually call the gene, right? And now we have the promoter region usually covering these sequence, usually certain sequences from the downstream and upstream, right? And to that region we know that RNA polymerase enzyme will come and bind into that region, right? We have the different subunits of the enzyme, and these multi subunit enzyme complex will occupy that promoter region, and that binding will be directed by the most significant factor called the sigma factor. So they help in the binding of this factor to the promoter site. So this is in precise what we have discussed in the presentation. So I hope you understood about this. So now we are winding up this session. So I hope you had an idea about the transcription, the different components involved in transcription mechanism, and thank you for listening and watching. And if you have any doubts, please ask, and we will discuss in the session.